That gives me my little countdown. Alright, so this is Lab 2 Part A, right? We've kind of split this one up into 2 parts because it's a lot of stuff. So we're just focusing today on this one exercise, examination of eukaryotic microbes, right? So today also is the only day that we're going to focus on eukaryotes. The rest of the semester is going to be the other cell type. Do you guys know what the other cell type is? Prokaryotes. Awesome. So some characteristics and some terminology that goes along with these. So I had this really long definition for you guys, right, of plastid on your homework already, right? And to kind of summarize, these are double membrane bound organelles. So these are only in eukaryotes, involved in the synthesis and storage of food. They're commonly found within cells that are photosynthetic organisms like plants. So this is related to what organelle you're familiar with in plants. Chloroplasts, right? Chloroplasts, right? But the evolution on how these came to be, right, you hear this terminology of plastid, which is where it kind of gets the name chloroplast. And we're going to talk a little bit about that today. Prokaryote, which is one of 2 cell types that we talk about, they're small, which with peptidoglycan in the membrane, if you're talking about bacteria. It lacks a nucleus, but it just means that it lacks the membranes that encase the DNA. So instead, it has DNA, usually circular, covalently closed DNA, within the cytoplasm, right? It's not sequestered into a membrane. It has ribosomal components, right? Remember, these are used to make what type of molecules in our cells? Proteins, right? All cells have to have that ability. So all cells have ribosomes. They're smaller, though, than eukaryotes, and they're a little bit different, but the functionality is the same. They tend to be what some people refer to as more primitive than eukaryotes, so not quite as complex. Eukaryotes are a larger cell type, and that's one of the nice things today is they'll be very easy to visualize under the microscope, most of them. And most of the time we'll see a nucleus, right? This membrane-bound area that contains the genetic DNA. It can be in linear strands and often containing introns, which is intervening sequences in the DNA. This is not seen in prokaryotes. So a little bit more complexity here, right? Hence why they consider them more complex. Ribosomal components, right, are similar but somewhat larger than prokaryotes. Although there is a belief that eukaryotes evolved after prokaryotes, so prokaryotes came first, right? And pro actually means before, right? Karyote refers to kernel, like the nucleus almost looks like a kernel inside a cell. Eu means true, right? So it has a true kernel, right? When we classify life, there's a classification scheme. There's probably one that you learned growing up. Kingdoms, phylums, classes, orders, all of that. Domains that we may be introducing new to you today are above kingdoms, right? And so this is a much broader classification. And this is actually based on nucleotide sequences of ribosomal RNA. So remember, all cells have to have ribosomes because they make proteins. So all cells have this ribosomal RNA component. And scientists were able to look at this, and when they compared all the sequences, they were able to break it up into 3 main groups. What we call bacteria, archaea, and both of these are prokaryotic, right? Both contain cells that don't contain a nucleus. Eukarya is the 3rd domain, and as the name implies, all of them are the eukaryotic cell type. So they all have a nucleus at some point in their life cycle. You should remember RNA, right? from biology class actually stands for ribonucleic acid. It tends to be a major intermediate, right? Unlike ribosomal RNA, which is important for building the ribosome, most RNA is messenger RNA. It is carrying that message between DNA and proteins, right? So you have your DNA in the cell that is transcribed commonly into messenger RNA, which is then read by the ribosome into proteins. This process is referred to as gene expression, right? When we go from that genetic information to actually making something like a protein. And proteins run your cells. They're the major component of your cells. But RNA, as I said, can also be structural, right? It's structurally important for ribosome. For the structure of a ribosome is ribosomal RNA and protein that actually makes up that component that attaches to the messenger RNA. And then in the process, I think I just ate a bug. Extra protein. I saw it fly. Oh, well. Okay. To transfer the amino acids to that RNA ribosome, messenger RNA ribosome complex, we have transfer RNAs, right? They actually transfer the amino acids to that complex and help us build proteins. So when I was a kid, I learned a mnemonic to help me know the classification scheme, the different groupings within the classification scheme. So, and Peter's got a different mnemonic in here that you guys maybe have heard, but the one that I learned when I was a kid was, Kevin, please close our front gate slowly. K for kingdom, right? Kevin, kingdom. Please, phylum. Close, class. Hour, order. Gate, groups, right? So notice for some of these, too, we even have classifications kind of in between, right? So for groups, because we have so many groups, they even have groups above groups, and they call those supergroups. So you may not have heard this terminology before. This is kind of in that, in the level there. So 5 divisions, right? And where are we? Kevin? Yeah, we'll look at it on the next, when I have the schematic. And so this is changing to that system that you may have learned when you're in biology class. The good news for this class is you're not going to be memorizing this schematic, right? But understanding that it exists, right? That we have classification for organisms. Part of the reason why I don't make you memorize this is not a phylogeny or classification taxonomy class. And these are kind of in an area of flux right now, a lot of changing happening. We want to maximize brain space and not stick stuff in there that you're not necessarily going to use, right? So even if I told you to memorize, you'd probably be like, I don't need to know that. So remember earlier I mentioned how we believe that prokaryotes and eukaryotes had this evolutionary relationship? One of the theories out there is referred to as the endosymbiotic theory. And that is a theory where we believe that primitive prokaryotes, right, so very early prokaryotes, were engulfed but not digested by a much larger prokaryote. And this led to the formation of what we now know as eukaryotic cells. So there's lots of evidence that back this up, especially with chloroplasts in plants, and this relationship with what we now know today as cyanobacteria. At one time they were known as red-green algae, but come to find out they're not in fact algae, they're not eukaryotic, they are in fact prokaryotic. So these cells are very similar to what probably happened. And what the belief is that you had this prokaryote, right, that had this ability to photosynthesize. It was engulfed by another cell that didn't have that ability. And so this cell began this symbiotic relationship, right, where they're both benefiting. So the one that's photosynthetic is being protected by the one that engulfed it. And then the one that's photosynthetic is providing energy for the one that engulfed it. right? So we have this partnership. And it gets to the point so much in evolutionary that they really can't live without each other. So they become one. All right. So cyanobacteria, when we look at them, they're very similar to the chloroplasts we see in modern day plants and algae and things like that. They have similar DNA sequences. They even have prokaryotic ribosomes, right? So there's another link of evidence that it may have been a prokaryote, right, before it was engulfed and began this relationship to where these two no longer can separate from each other. Right? A plant cannot live without its chloroplast, and a chloroplast cannot exist on its own anymore, right? They've evolved together, so they've lost their ability to be without each other. Does that make sense to you guys? This is also believed to be true for mitochondria, right? That they were once a prokaryote all by themselves and then became an organelle within eukaryotes. Some of the organisms we're going to look at today have different phases in their life cycle. So we're going to focus on looking at under the microscope because they're much more interesting too and easier to identify. Any organisms that have a trophocyte. So you'll notice for some of your slides it says a trophocyte. And so this is the feeding phase within the life cycle of some protozoa. Other protozoa have the ability to form a resting form. So just as it says, they're not feeding, they're just resting. They tend to be more resistant to harmful environmental conditions. So there's sometimes a phase in their life cycle where they need to be protected in order to survive. And this would be the cyst. These are much harder to see and identify under the microscope. So although we'll talk about organisms that have this ability, we won't look at it. How organisms feed and eat. We've already kind of mentioned, right, how some are photosynthetic. Others kind of have to eat like us. They have to engulf other once living or living organisms. Autotroph is a specific term used to look at how they're acquiring certain components. And autotrophs refers specifically to where they get their carbon from. So some organisms have the ability to get their carbon that builds the building block of all their molecules from carbon dioxide. And that's a lot of your photosynthetic organisms are actually incorporating carbon dioxide and using light energy to put that together to form their molecules. But some other organisms, some prokaryotes, some bacteria and archaea can actually utilize carbon dioxide. We don't utilize it, we produce it, don't we? We produce carbon dioxide. We cannot take carbon dioxide and form molecules with it. We don't have that ability. We're the other, what's referred to as heterotrophic. So we require our carbon be in an organic form, meaning some other organism already put it together for us. And plants are the major ones that do this, right? And then animals eat them. And in the case of us, most of us are omnivores. Some of you might be vegetarians or vegans, right? So you're eating plants that preform carbon for you, right, in an organic form. Or you're eating plants and animals, and of course most of those animals you're eating, where do they get their energy from? At one point they eat plants, right? Some of them are omnivores like us and eat plants and animals, but at the very basic level you have animals that eat just plants, right? Even some humans. And so you can get that organic source of carbon. Mixotrophs, mix means more than one, right? You're mixing things up. So these ones can live autotrophically, and usually, again, they're using light energy to do this, to use carbon. But sometimes light isn't available, right? So they have the ability to switch to organic sources. Lots of prokaryotes have these abilities because as the environment changes, if you're not able to adapt to that, then you're going to die off, right? So there are organisms out there that can actually switch it up, right? They're not stuck. Like us, we're stuck. We're heterotrophs, right? There's no way we can do autotrophic, right? That'd be a really interesting evolution, right? We'd probably look like green Martians and we could tell our moms we don't have to eat vegetables anymore, right? Because we would be vegetable. We would be plant matter to a certain extent. Pseudopods is an interesting ability that some microscopic eukaryotes have the ability to move. And it's sometimes referred to as false feet, right? Pseudo meaning false, pods referring to projections, feet. And so this is very common in amoeboid-like organisms. So hopefully in biology class you may have seen these before. There's lots of cool YouTube videos out there of these moving. I've even taken some videos in class. So unfortunately, because of this, these guys tend to have a pretty irregular shape, right? Because they're always constantly changing their shape in order to move. They actually use this for feeding as well, right? So they can surround stuff with their cytoplasm, with their membrane, and engulf it and take it inwards. And again, lots of cool pictures on YouTube out there of this actually happening. Cilia is little tiny fibrous projections. They typically move in a whip-like fashion, and a lot of times the organisms are completely covered in them. They not only use it for locomotion, but they also can use it for feeding, right? To move the food towards them into an area, into a groove where they can then engulf it. So paramecium is one that most beginning biologists are familiar with that look like this, that have this particular organelle. This is unique to eukaryotes. Saprophytes, again, has to do with how they feed. These are usually heterotrophs, again, so organic sources of carbon. But in this case, we're referring to that as being dead. And these are really important organisms because they're our decomposers. A lot of your fungi, your molds and such, are saprophytes. They're feeding on stuff that's dead, right, and recycling it back into the environment. A parasite is obviously usually something we don't want to be involved with because that relationship is harmful, right? So it's an organism that lives symbiotically, right, has to live within or on another organism, but the organism that lives on or within that host is harmed by them, right, and can eventually sometimes be killed. So we always think of bacteria as parasitic, but worms, right, some of the protozoa that we'll look at today can be parasitic, that bad relationship. Molds is a general term, right, for filamentous fungi. Most of us have seen mold at one time or another growing on our bread, right? So those really fine fibers we'll actually look at under the microscope. That's why they look fuzzy, right? It's those filaments that it's made of. Yeast, on the other hand, looks different. Sometimes growing on a surface, you wouldn't really denote them as being any different from bacteria. But they are, in fact, unicellular, microscopic fungi. And we'll look at those again today under the microscope. So when we talk about hyphae or hypha, right, we're either talking about hypha, just a single filament, or hyphae, multiple filaments of focal cells. And this can only be seen microscopically to significant detail. Mycelium is that mass that you would see growing on your bread, right, or on an agar plate. So not at the microscopic level. A lot of these organisms, what you're seeing growing up is its reproductive structures. So for instance, Aspergillus, those fruiting bodies, sometimes referred to as, these are structures, and the spores themselves are going to be arranged in a particular way at the end. So, especially for Aspergillus, kind of looks like, remember dandelions when they have that ball, right, for their spores, for their pollen? Aspergillus kind of looks like that as well. And then when we look at Penicillium, its spores are going to be arranged a little bit differently. Almost looks like brushes. It's actually where it gets its name, brush-like. And then, so that's asexual reproduction, typically when they're forming these aerial fruity bodies that we refer to as conidia. Zygospore is a little bit different. This is sexual reproduction for moles. So mitosis is happening, meiosis, instead of mitosis. So you're getting two different ones coming together. And we're going to look at a particular type of zygospore. And this is within the hyphae, not the aerial hyphae. So this definitely brings you to our outline, right? In that we have the two different cell types that we've mentioned, the prokaryotes and the eukaryotes. And in this picture you see both. So the prokaryotes here are the purple little dots that you see, probably bacteria. And then, I think this is actually a swab from someone's mouth. You see the big pink cell here with the darker pink center? That's a eukaryote, probably is a cheek cell. Okay? A squamous epithelial cell for any of you guys taking A&P. Right? So, here's one, and here's probably another one. Right? Another large pink cell with the nucleus here. So as you can see, as we've said, they tend to be much larger than the bacteria. So this classification system that I was kind of mentioning, right, has been going on for a really long time. Linnaeus is actually one of the ones credited with the father of what we call taxonomy, this organizing, classifying organisms. So, handsome devil there, back in 1735, published Systematic Nature, right? And so classifying organisms into these things that we're used to hearing, right? Things like classes, and genera, and species. So here's that classification scheme, right, that I talked about. Kingdoms, phylums, classes, order, family, genus, species. And then the addition of this one at the top of domains. That's probably relatively new to you guys. And we're going to focus on what domain today? So there's bacteria, archaea, and eukarya. Eukaryotes, right? So remember the saying I told you guys, Kevin, please close our front gate slowly, for remembering this order. So if you needed to remember that order and we need to add in the D now, you could say, damn, Kevin, please close our front gate slowly, right, for our D for domain. But as you can see, right, as we go down, right, we could get down to a single species. How is this classification determined? I mean, what makes an animal an animal? What's a chordata? What's a mammal? What was this information based on? Right? We know bears, right? Because they have similar what? Characteristics, right? So we would look at traits, right? Or what we call characteristics, right? Having hair is a mammalian trait, right? So notice the snakes are not included in that grouping. Chordatas have the spinal cord, right? And then we get up to other animals, right? That kingdom, notice we add in things like starfish, which don't have a spinal cord, right? But would be considered part of the animal kingdom, right? As we go down, right? Carnivores, well, those are ones that eat meat. We knock out the squirrel there, right? As well as many others. Squirrels don't eat meat, right? What do they typically eat? Nuts. That's plant matter, right? Okay. And then we have the different types of bears, right? And then down to a single species of bear. I think this is the brown bear. Unlike the black bears, yeah. Oh, grizzly bear they have here. Ooh, those guys are really scary. I'm glad we don't have those here. Right? So, what domain are you guys already answered that? Right? Is eukarya. Which includes all the ones that we've looked at, right? All those animals on that chart. So, in looking at that, there's some problems sometimes when you just look at characteristics, right? Whales, sharks, and fish, are they all the same? They have similar characteristics, right? What are some similar characteristics? They live in the water. They have fins. Oh, gills. Not all of them have gills, right? Whales don't have gills, right? They breathe air, right? Fish and sharks do, right? So there's a difference. We separated out the whales from that, right? And so remember earlier I talked about a mnemonic for remembering. Here's that other one. King Philip, come over from Germany swimming. For kingdom, phylum, class, order, families, genus, and species. I always like the Kevin close our front gate slowly. I don't have any trouble remembering that one. So pneumonic plague, though, is something completely different, right? That's an infection caused by a bacteria. It's pestis. Make you really sick. So taxonomy is a classification word into categories, right? So we're grouping organisms. Phylogeny studies the evolutionary development as well of species. And the way that we've done that a lot of times is adding in genetic information like the ribosomal RNA to some characteristics. Ontology is the course of development, right? So how an organism develops. And you can see there's similarities in development of organisms. Like, for instance, in our development, we actually have a tail, but we lose it, right? So like other mammals like cats and stuff like that have tails, right? We do too, it's just we lose it in our development. We have it early on in development and then we lose it. Why that is, who knows? But I'm actually kind of glad I don't have a tail. So it may help us, right, in this understanding of classification and evolution, and even looking at the development of organisms, not just when it's born what it looks like, but how it develops. So as we said, whales and fish have similarities, right? They live in water, they have fins, they can swim. But whales have hair, just like us, because they're mammals. They give live birth, like us. And they require O2, right, for breathing. Where fish, on the other hand, have scales. They do not have hair. They lay eggs, right? And they acquire their oxygen from the water itself through gills. So, really, depending on how you classify things, sometimes you can almost misclassify things, right? So, there's a lot in the science of classification and phylogeny, and a lot of stuff going on. Phylogeny tries to take more into account the relationship over time, right? So almost like a family tree that we're trying to deduce, right? And a lot of times using genetic information. You wonder why you look like your parents. Well, because genetically you are your parents, right? Just re-scrambled. Okay? So you have this evolutionary relationship back through time, right? Some of us can even look at our grandparents' pictures and be like, wow, I look a lot like him or her, right? And again, it's because genetically you're linked, right? Through time. So when we look at these interactions, right, we believe way back in the beginning it was some prokaryote, and then we had this branching off between bacteria and archaea, and really some of them even potentially coming back together, right, in that endosymbiotic theory of one being engulfed by another and getting our eukaryotes. And then even then, the interactions between them back and forth led to other branches, right? So the beginning, they believe mostly our protista are our very beginning, our protozoa and stuff like that. And then the ones that were photosynthetic branched off into the plant kingdom that we're used to looking at. And then other protista went more towards decomposition and the fungi that we see in modern day. And then the other branch of animals, right, where we mostly went through heterotrophic means of acquiring organic material. So it isn't quite the straight tree that most people envision. So as I said, in the domains, right, we have these groupings of eukarya. These are all the eukaryotic cell types. And for prokaryotes, we have archaea and bacteria. And we're mostly going to stick to bacteria, that's what we know most of in this course, right? But don't forget about these other prokaryotes that are new on the scene. Part of the reason why you don't talk too much about them in most of your classes, most of you guys are allied health oriented, they found maybe one or two organisms that are pathogenic to humans. So hence why we, as allied health and health related individuals, why you don't hear so much about archaea, right? Because they don't directly affect health. But they do affect our life on this planet, right? and the important roles that they play. Otherwise, they wouldn't be here anymore. They'd become extinct. So remember, here we are under animals, right? Humans, we tend to put ourselves on this pedestal, but you are an animal, right? So we are eukaryotes, but multicellular, right? So we're going to be looking at the small stuff today. So supergroups, right, come under the domains. And as I said, I'm just making you aware of this classification. By no means do I want you guys memorizing this. But you'll see, right, some of this terminology, and that's what they're talking about, where it's grouped in the whole tree of life for animals. So we've shrunk down bacteria and archaea here, and we're going to start out in this supergroup that even in this tree that I found breaks it into two other main branches. And Unicodata. It's fun to try and say some of these names, huh? So we're humans. We are in Metrozoa, actually. And Berkeley has an awesome website, which I put a link to for you here, of all the Metrozoa. So going back to our schematic, right? So for prokaryotes, remember bacteria, which is going to be the main focus of this course after today's lab, and then archaea. Just one domain has eukaryotes, and that's our focus for today, and that's eukarya. Within that domain, we typically still go with a lot of the kingdoms, right? So plants, animals, protists, and fungi. Protists are typically broken into what's referred to as protozoa and algae, where fungi are commonly broken into yeast and molds. We're going to stick to these two classifications because this is a microbiology class, right? We're not going to look at plants and animals because they're not microscopic. So then we go into the many different subgroups that you saw on that tree. So notice this is important, right? This breakdown. So we're going to start out in excavata. In the Basilidia. So these are some pictures of the organisms that would fall under this group. Anyone recognize this one from the homework? Giardia, right? Almost looks like a little animal itself. Almost looks like it's smiling at you. These are not eyes though, right? What are they? Nuclei, paired nuclei. Euglena, probably something you've seen in the past, right? That can swim through pond water. It's kind of green, has photosynthetic ability. So this grouping of excavata, again, similar characteristics. They're all going to be unicellular. They're all going to have a feeding groove, usually, whether you'll see this or not. Depends on the specimen you're looking at. They tend to have one or more flagella. So these are long protein-like appendages that are going to allow it to swim. And again, a lot of these have that trophocyte. So remember, what is that? The feeding stage, where the cyst is the resting stage. So we have 4 different subgroups and 4 different organisms. And unfortunately, we don't have slides that are good for any of these guys. And so these are all parasites, though, except for Euglena, that if you got infected with them, you would have to take antiparasitic drugs. So Trichomonas vaginosus, as you can tell by the name, right, women probably want to avoid this one because it can infect the vagina, as the name implies. You can see the nucleus, right, which again clues you in that you're probably looking at a eukaryotic organism. You can see the flagella here. These guys are really cool. I wish we could get some live specimens that are non-pathogenic because they have an undulating membrane, kind of waves. Only the trophocyte form is known. They're not known to have a cyst form. They're a fermenter, so they can't do oxidative phosphorylation like we do. They have hydrosomes, so they produce hydrogen gas under anaerobic conditions, which is really interesting. And they can cause sexually transmitted disease. Anyone know which one? It's commonly referred to as trichomonas, or commonly referred to as the trick. Just trick. Yeah. It can also cause an infection not only of the vagina, but also of the vulva for women. Sometimes it's confused with other organisms. There's a roundworm, trichinella, that can cause an infection in this area, as well as a whipworm. So when someone has the trick, you don't necessarily know which one they're talking about sometimes. So an additional information would need to be taken to try and pin down what type of organism. It's believed to cause a problem when people have disruption in the normal flora, when your bacteria and such like that are disrupted.
So women get it from men?
You can potentially get it from men. And as you can see, the infection is quite complex, and we still don't really know exactly what's going on.
But we know how to treat it, sort of.
Yeah, if we can figure out what it is you have, we can treat it. So Giardia labia, a lot of people actually infected with this one. It's actually referred nowadays as Giardia instantalis, although I think they have labia in your book, right? Yeah. And again, because it can infect the labia for some individuals. Most commonly, it's found in the intestines. So you can see, even in our book, we have a really crappy picture, right? And you can't really see the flagella that are associated. There's actually 4 pairs of flagella with this organism. As I said, we don't even have a good slide to look at, unfortunately. And this is the trophocyte. In addition, they had non-functional mitochondria, so they must rely on glycolysis. These are referred to as metrosomes. Paired nuclei, right, which kind of look like eyes. The flagella. 2 medial bodies that you can see here, almost looks like the smiley face sometimes. They can form cysts that are resistant to water treatment. So this is the bad news, is when they get into our water supply, you could ingest the cyst. The cyst can germinate in your intestinal tract, attach, and cause pretty serious infection. So found in streams, lake waters, especially stagnant water such as ponds, formed by beavers' dams. So sometimes this is referred to as beaver fever, an infection. As I said, most common in the intestines, although some people can get it for women, can get it in the reproductive area. Explosive diarrhea, malaise, excessive gas, often flatulence or a foul, sulfuric tasting belch. Gross. Although there's a lot of people too that have this infection are asymptomatic, don't even have this problem. Good old Anthony von Leeuwenhoek, one of the first people who contributed with seeing microorganisms because he wrote about it to the Royal Society of London. He actually looked at his own poo, and he's also credited with seeing the first, probably, Giardia. Yeah, usually self-limiting, rarely is it fatal. So looking at these guys are super cool organisms. They're green because of their photosynthetic pigments. They tend to have 1 or 2 flagella. They have an eye spot known as the stigmata, so this allows them to move towards light in the water column to help them photosynthesize. But they're mesotrophic, so they can do light, utilize light energy when it's available, but they also can be heterotrophic, engulf other organisms for energy when light is absent. So these are one of those ones that was like, well, is it a plant or an animal? Well, it does a little bit of both, right? It's in that weird in-between category. So what structure gives them their photosynthetic ability? That would be their chloroplast. And again, we don't unfortunately have any slides of these guys, but you've probably seen them. We've got some pond water. We can try and make some wet mounts. We might get lucky and see them. Trypanosoma. These guys have one enormous mitochondria. Anyone know what disease they can cause? Right, they can cause African sleeping sickness. So the good news is here in the United States, you don't have to worry about that. We don't have this parasite, this protozoa. It's transmitted by the bite of a tsetse fly, actually, in Africa. And there's two different subspecies of it. But there are other Trypanosoma cruzi, which can cause Chig's disease throughout South and Central America. Again, thankfully not here in the United States. So this Trypanosoma is a group name, right? But these are two examples of two different genuses, the brucei causing the sleeping sickness, and the cruzi, which can cause Chig's disease. Actually had a colleague get this on his travels. So that finishes up Exterava, and unfortunately we won't really look at any of those under the microscope, which is a bummer. The next group is Archaeoplastida, and since it has the plastid in its name, right, kind of gives you a clue that we're going to be dealing with things like chloroplasts, especially when we're looking at green algae, which are chlorophytes. And there's a really cool one, and unfortunately we won't see it to this level of detail. This is a single cell that lives within a colony of cells known as volvox, right? And so this is the individual cells that make up each one of these little green dots. And then even in here, multiple green dots making up this colony, right? So it's a group of them living together. And so they form colonies within colonies. So we'll see that. So here's a colony with little colonies inside of it, what we call daughter colonies. So this is the level of detail, unfortunately, we'll see under the microscope today. But if you were to look at an individual cell, this is what they look at like. And you can see they have flagella. You can see they're greenish in colorization. Unfortunately, the ones we have today have been stained, so you won't see the green. You'll see whatever they were stained with. And so as you can see, they form, groupings to form that colony structure that you see here. Really cool organisms. And then we have chlorophytes. This is another grouping, and again, you can see the chloroplast here, and this one's really easy to identify because the chloroplasts are actually within the cell, and this is just a single cell. They form filaments. Those are the chloroplasts that are spiral shaped. So this is our spirogyra that we'll look at today. And sometimes right in the middle of a cell. So this is the cell wall between cells. We'll look and hopefully find some nucleus. So what do all these guys have in common? Chloroplasts, right? Remember I said most of them are photosynthetic, are also autotrophic. So what are they using as their carbon source? Carbon dioxide. Cellulose makes up their cell walls. And this is actually a sugar compound, glucose. But the way the glucose are hooked together, it forms this really structurally strong cell wall. We have glucose in our bodies too, but not organized to this level. And we actually don't have the ability to digest this compound. Bacteria do, right? And so when cows and such are eating it, it's actually the microorganisms in their gut that are digesting the cellulose, not the cow. And we can't digest it, so it goes in one end and out the other. So when you eat corn kernels, right? Corn is actually a single cell embryo of a corn plant, right? It's the egg that you're eating. And so if you don't bust that outside covering, which is the cell wall, the cellulose, it goes in one end and out the other, right? You ever have whole corn kernels come out in your poop? It's because you didn't chew your food. Chew your food. You've got to bust open that capsule, otherwise you don't have the enzymes to break it. So then you just wasted it. It just went right through. You got no nutrients from it. Right? When they talk about fiber, that's what they're talking about is cellulose, because guess what it does? It goes right through. Right? Because you can't digest it. But it helps move stuff through. Right? Fiber helps move stuff through. Right? But you don't digest fiber. You don't digest cellulose. Right? Some organisms can. Right? We do not. So the chlorophytes, we usually will see those in fresh water. They're unicellular or filamentous or colonial, right? As we saw, they can form colonies or they can form filaments. And we'll look at those guys under the microscope. The Volvox. Chlamydomonas we don't have. This one sometimes we'll see in some of our live cultures as food for our amoeba and paramecium. We do have the Spirogyra. And I have this awesome thing that I found on the internet for Chlamydomonas. These guys are really unique, little single-celled, and again, they swim around like crazy. They look like little balls running around when you see them in culture. Right here I have a link to this funny little song that this guy Andy did. If you have time, click on the link to it. It's so cute. Not to confuse with gram-negative bacteria, right? Chlamydia trichomatis, which causes the sexually transmitted disease chlamydia, right? So chlamydomonas, chlamydia, different, right? Sounds similar, totally different. And good old conjunctivitis, which can be caused by bacteria or viruses, not caused by these guys. And then Neisseria gonorrhea, another bad sexually transmitted disease, right, commonly referred to as the clap, is again a bacterial infection. So here's our Volvox, right? We will look at these guys under the microscope. So again, individual cells you can't really see in this great of a detail form colonies, and then even smaller colonies within a colony that will eventually leave this colony and form their own. These are called daughter colonies. So how are these produced? What are daughter colonies? What type of reproduction? Asexual. Right? Asexual reproduction. Where are we on our list? Yeah, asexual. So you notice these questions that we're answering on here? I would highlight them, right? Because these are testable, quizzable questions. Asexual. So this is an excellent website that has really cool pictures too if you're really interested in Volvox. So lots of references here for you guys. Again, we got the Spirogyra. You probably looked at this one before. These are filamentous. These are haploid, just one set of chromosomes. They reproduce by conjugation. We got a couple of conjugation slides where you have two coming together and exchanging genetic information. That's not happening in this particular picture. And they'll form a zygospore, right? So that's sexual reproduction. So we'll look at those. And here's the nucleus right here in the center. See how it's quite distinct? And then again, those spiral organization of the chloroplast. Oh, there's mine. So this is that zygospore. See how two came together? And it formed that sexual spore that will bust out of there and form a whole nother genetically different strand of Spirogyra. So what are we going to need to be able to identify those spiral-shaped chloroplasts, the nucleus, and again, notice the division between cells, that these are filamentous. That's the cell wall. You can really see the cell wall there between those guys. Okay, our next grouping are sometimes informally referred to as your ciliates, because as you can see, they're covered in cilia. See all those little hair-like appendages? Right, especially on paramecium. Unfortunately, don't, because they're so tiny, it's really hard to see under the microscope. Sometimes if you focus up and down, you can see the fuzziness to them. And we don't have all of these specimens, unfortunately, but we've got quite a few of them. So we have the ciliates, and we're going to also look at the diatoms. These are my favorite. And this is actually a ciliate ingesting diatoms. Right? So organisms eat each other, right, in these environments. This is really cool. This is an algal bloom where they have fluorescent bioluminescent ability, right? And some of you guys may have seen this on the news not too long ago. So little tiny organisms, dinoflagellants that have this ability. Pretty cool stuff. So this is what I was talking about before too, right? How we believe that a pre-algal cell ingested a cyanobacteria. They began a symbiotic relationship, and then even some of them even were ingested by another organism, where you get these organizations, these plastids that are generating energy, typically in this case from light. So they believe this happened twice within this grouping, right? 2 engulfments happened. So for this one, we'll look at paramecium. We're going to look at, we don't have any centaurs, unfortunately. Those guys are pretty cool. Those are those trumpet-shaped ones. Lots of YouTube videos and stuff out there for those guys. They're really cool to look at. And Bellatium coli, we do have that particular ciliate to look at. And then within the non-modal ones, we have the Plasmodium falciparum. And what disease does this one cause? Malaria. This one causes malaria, transmitted by mosquitoes. Thankfully, we don't have this particular protozoa here in the United States. At one time they did, right? But we don't anymore. And then we have our diatoms. Most have a unique flagellum with 3-part lateral hairs, right? So these guys are modal as well. So in our ciliate grouping, these ones with cilia, we have the paramecium, and our slides don't even look quite this pretty. We don't have the specially stained ones where you can see the cilia. But as I said, if you focus up and down, you can kind of see the fuzziness around the edge, right? And they're completely covered. This is a 3-dimensional picture. What we're going to be looking for in these guys is the macronucleus. You should be able to see. If you see a micronucleus, I'll get really excited. The cilia, again, is really difficult to see, but you still need to know that these guys have cilia, and that's how they move. We may be able to see their oral groove. Again, this is much easier to see when you have a live specimen. And the contractile vacuoles, if we have live ones, you can see them squirting the water back out. It's like pretty cool stuff. And lots of, look, I even got links to cool YouTube videos, and you can watch that happening. Alright, and here's the pretty centaurs, those trumpet-shaped ones. As I said, unfortunately, we don't have these guys. And you can see they have beaded macronucleuses. Kind of cool stuff. A white spot in the dark coil. Not sure. The cygromes. They're naturally green. This is one of the ones we will look at. Again, a ciliate. And this one has a trophocyte and a cyst stage. We're going to just look at the trophocyte. This is what the trophocyte looks like. Again, you'd see cilia along the edge if you focus up and down. They have a very unique macronucleus, tends to be kidney bean shaped, where the cells themselves tend to be oblong or round that we'll see. What symptoms would you have for an infection with one of these guys? Where am I spotting that one? Oh, okay. Yeah. Bloody mucoid stool, diarrhea, often as early as 20 minutes after ingestion. Chronic infection, alternating diarrhea and constipation an individual could experience. This occurs more commonly in developing countries, right, where they don't have good, safe water supplies, like the Philippines, where the water can be contaminated by swine or human feces. It's one of the few ciliates, right, known to infect humans. Right, you don't have to worry about paramecium making you sick, right? But this guy you would. The Plasmodium falciparum, and this is just one species of 4 different species of Plasmodium that can cause malaria. And because of that, you'll notice on the slides we'll see red blood cells, human red blood cells, and then you'll see the parasites. The parasites themselves are sometimes inside the red blood cells because this is part of their life cycle that they reproduce inside the red blood cell, and then part where they'll be outside the red blood cell. So you need to look in both places in order to find them. And as we said, this one causes malaria. And so here's the life cycle, right, and additional information about the different morphology you may see and the different parts of the life cycle. But on those slides, you're either going to see red blood cells or the parasite. So if it's not a red blood cell, it's the parasite. And you guys won't have to identify what phase of the life cycle it is. It's too difficult. So mosquitoes and ticks, as we know, transmit lots of different types of diseases. So these are some additional diseases that you may hear of, right? Things like yellow fever, right? Different types of mosquitoes transmit different types of disease as well, right? We think of mosquitoes just as a single mosquito, right? And we do have the ones here that could transmit these diseases. The good news is we don't have the viruses that cause these infections here to be spread. West Nile, on the other hand, we do have to worry about, right? And it's mosquitoes. And then good old ticks, right? Transmit a lot of different types of diseases as well. So Plasmodium falciparum, right? Where in the world would you worry about these types of things, right? And so you can see mostly in Africa, South America, even Central America, India, different parts of Arabia, right? Arabia Peninsula, Philippines. So there's lots of information. Remember I said we went overboard? So you can look at lots of information. So that brings us to our diatoms that we're going to look at. These are sometimes known as nature stained glass because what type of substance is made up of their cell wall? Silica. Silicon dioxide. This is what glass is made out of. Hence why they tend to be as pretty as glass. A lot of them are photosynthetic as well. Common shapes that you'll see. Right, because they can refract the light, they do have a brownish type pigment usually, but you'll see them in lots of different colors. These elongated forms are referred to as having bilateral elongated, or what we call pinnate symmetry. It's actually two halves that come together. These guys are almost like petri dishes. Centric, which you see here, is a circular or radial or spherical symmetry to them. Of course, they just look circular to us under the microscope. So you need to be able to identify the two different types, right? So this is elongated, this is pinnate, this is centric, right? This is what they look like. Here's a centric one here. There's another centric one next to it. This picture, oh, that's just a zoomed in on a centric one too as well. There's an elongated one. Believed to have been evolved from red algae. So these guys are photosynthetic, right? Autotrophs, use carbon dioxide. They do have flagella. We won't see those, too tiny to see under the microscope. And then that brings us to the amoeba, right? And this is Entamoeba histolytica. So again, with the amoeboids, they're moving by those pseudopods. So they're not going to have a characteristic shape to them. And but these guys have a unique nucleus that when scientists are looking at poo, right, because this is an infection you could get, they'd be looking at fecal matter, they have a very distinct nucleus, sometimes described as what? Almost looks like a bullseye, right? And it's circular and then it has a dot in the center. Here's an amoeba, right? Here's long pseudopods coming out, and they're actually using this to probably engulf this food. So again, they're not going to have a very characteristic shape. They're going to kind of look like the blob. Here's an amoeba. This is actually an artist rendering, but it's really cool. An amoeba engulfing a paramecium. Kind of cool. Lots of videos out there, right, of this. So this is actually a picture I took of some of ours in the lab that we watched moving. But we're going to have stained specimens, not quite as good as this one. But again, you should be able to clearly identify the nucleus and pseudopods, right? These projections sticking out. Proteus is actually named after the Greek god of the sea, Proteus. Amoeba Proteus. So these guys live in aquatic environments. No cell wall, otherwise they wouldn't be able to morph their shape, moved by those pseudopods. Lots of links. Right? You can watch them move, watch them feed. Entamoeba histolytica. This is the one where and you can really see it in this picture, right? That bullseye-shaped nucleus. The ring with the dot. These guys can cause pretty serious dysentery, diarrhea. And so we would look at a fecal sample. And that's what we have, and so there's a whole bunch of other gunk on the slides. And when you're talking about an organism that doesn't have a characteristic shape, it's like impossible to find them under the microscope. So if you really want to challenge yourself, I'll give you a couple slides to look at. Alright, so that brings us to our fungi, right? So these are some pictures of yeast, right? And then the longer filament forms. So for fungi, they're non-modal typically. They have cell walls made of chitin, so this is a different polysaccharide, different from cellulose, but again, we're talking about sugars hooked together. There are different types of sugars. These guys are absorptive, which means they excrete their enzymes into the environment and then suck up their nutrients. Unicellular fungi, collectively known as yeast, they have a vegetative, actively metabolizing form, and then they bud. Little buds come off. This is how they asexually reproduce, and it's called budding because they literally look like a smaller bud coming off of it. And so this is uneven cellular division that happens. This is mitosis. So here's a really good picture of that. So here's a cell with a smaller bud coming off of it, right? And this is probably an earlier bud. It's a little bit larger. And then sometimes these buds will stay and elongate. This is not hyphae, right? This is what we call pseudo-hyphae because it almost looks like long filaments, right? But they're actually the budding cells. They stretch out, and so we'll see this characteristically with one of the yeast that we look at, right? You'll see these long threads, and you almost think you're seeing hyphae. You're not. You're seeing these spores you'll see these buds as they elongate. Sorry, I mean to say spores. I don't know why I want to say spores. Where molds themselves are multicellular and they are filamentous. So an individual filament we refer to as the hyphae. Hyphae is several of them, right? And that collection, that entanglement that you would see, that feathery looking, is the mycelium. Right, so that whole big mass of it together. So, and this is, you can see all those filaments, right? See all those lines, right? That's lots and lots of hyphae. Where you can see individual cells here for our yeast. So their classification is based on their sexual spore production and on the presence of cross walls in their hyphae, which we're not going to look at in the specimens that we're looking at. So there's two different yeasts that we'll look at, Saccharomyces cerevisiae and Candida albicans. Canada. And then there's three different molds we're going to look at. We're going to look at Rhizopus, Aspergillus, and Penicillium. And again, we can tell their difference by their organization of their reproductive structures, the different genera. So I think in your book, they have a picture for one of the yeasts that kind of looks more like the other yeast, and that confuses some of you guys. I think they have a picture like this and saying it's Candida albicans. And it very well could be, okay? But it's really not showing the characteristic pseudohyphae that one tends to have. And it actually, its picture looks a lot more like the Saccharomyces that we see. So I have an additional picture for you guys. So the vegetative cells, sometimes you'll even see the nuclei stained depending on the specimen. And definitely look for budding cells. Like for instance, this one you can see the vegetative cell and then the two buds coming off, right? You really want to try and see that budding that happens for these cells. Does Saccharomyces cause infection in humans? No. This is the one that makes, bread, mold. This is the one commonly called baker's yeast or brewer's yeast. This stuff makes beer, bread, and wine. Beer and wine, the stuff that we like, right? This is one that's a good yeast, you could say. One we actually use for manufacturing. So, Canidia albicans, it does bud, but its buds tend to elongate. And so this is a picture actually from one of our slides. You see those long filaments? This is not hyphae though, this is pseudohyphae. Alright? And this organism actually does this in response to temperature. So it's not exactly wrong, that picture in your book, right? At 30 degrees, it's going to be budding like this. But when you grow it at 37, you'll get those elongated hyphae. And this is typically how it would be for our body. Is this organism an opportunistic pathogen? Yes, this is the one that commonly causes for women what we call yeast infections of the vulva or the vagina. Can also cause oral cavity infections, commonly referred to as thrush. Can also cause infections of the skin. This is more common in people who have compromised immune systems, right? Elderly, AIDS patients, people going through chemotherapy. But anything that can cause a disruption in the microflora for women, antibiotic therapies, hormone changes, some diabetics because you have more sugar and yeast loves sugar, you're more apt to these types of infections. And so some more interesting information on those. So that brings us last to our molds, right? So as I said, most of us have seen it growing on our bread. Good old fungi. So we have 3 different species that we'll look at, and we'll be looking at their reproductive structures to be able to differentiate them. And for rhizopus, we actually have the sexual and asexual spores. And so as you can see here, it's pointing, see how the 2 hyphae are coming together? That's a zygospore, that's a sexual spore. This is an aerial hyphae, and the spores are arranged in a particular way. And I've got some better pictures. And then the threads, right, those filaments of the hyphae. So lots of hyphae seen here. The aspergillus, remember I said, kind of looks like the dandelion when they spore. And here's some individual spores. Penicillium is my favorite because their spores line up on these long projections that almost look like brushes. And actually, penicillium actually means brush-like. So under the microscope, right, this is actually a slice, and I have this slide where you can see under the microscope the very thin microscopic filaments that we call hyphae. And in this section where it's dark, it's actually in the agar. This section is where it's growing up above. So I took a plate like this one you see here. You see these fuzzy fungi colonies that we call mycelium. Actually took a slice, right through the agar and right through the top, and put it on a microscope slide, and I stained it with methylene blue so you could see the actual fibers. So as I said, when you look at this, there's going to be a section where you can see in the agar, and you actually can see it growing up out of the agar. So when this stuff is growing on your bread and your cheese, it grows up, but it also grows into the cheese, right? And it's putting its digestive enzymes into that, right, to digest that food. So for some that are pathogenic, those enzymes can diffuse through the whole block of cheese. So just cutting off that section that has the mold on it, not a good idea. Or the bread, right, just toss it. Most of them aren't going to harm you, and in fact, aspergillus, right, and the picture they have in your book of the spore structure is actually been sliced We don't have any fancy slides like that. Ours is not sliced, and so it's that ball, like I said, right? So almost like that dandelion ball. They wanted to show you the supportive structure in the book. That's all well and fine, but those are expensive slides. So these organisms are actually somewhat good. They actually produce a commercial product that we actually take from them. Did you guys figure out what that was? Yeah, citric acid. It's also used to make soy paste and soy sauce. Right, from soybeans. It's also a major component of mildew. Used for lots of things. Penicillium you probably know by its most famous byproduct that produces under certain circumstances, and that's penicillium. It's an antibiotic. It's a chemical that it produces to affect bacteria, right? And we basically just stole their technology, right? We steal that chemical and put it in pill form. But this is where it comes from. And so as I said, these guys are really cool because their spores kind of line up in these columns. Almost look like bristles of a brush. And in Latin, actually, penicillius actually means paintbrush. It's actually where it gets its name, is based on its structure. So one of the first antibiotics discovered, the organisms only produce this under stressful conditions, right, when they're potentially having to fight off bacteria for food and environment. So this is actually utilized in making some food products, which are good old blue vein cheeses, right? Blue cheese is the most common, but Cabernet, Brie, other cheeses actually use this fungus to make it, to ferment the milk products to produce these cheeses. Although if you have a penicillin allergy, you can still eat them. That's because they're not producing penicillin under those conditions, right? They only produce it under stressful conditions. So we actually have to put them under particular stressful conditions to get them to produce penicillin to be able to harvest it to make penicillin. So there's multiple good aspects. It's actually used in a lot of fermentation processes in many sausage and ham productions, as well as other molds and bacteria. So microorganisms are actually quite beneficial to us. Rhizopus, this one commonly you're going to find where? On your bread. This is the one you typically are seeing at home. And if you look at it microscopically, it's asexual spores arranged in this like cap-like structure on top of the aerial hyphae, the conidia. And two hyphae will come together to form that sexual spore, and this one's referred to as the zygospore. And that's it for our lecture. I'm going to pull up that picture. What did I do with it? Alright. Remember, go to Blackboard for the videos for next time. Make sure you watch those so you're most familiar, and I will demo. And here is our instructor IT sheet. I made this available to you guys as well. Right, so we're going to actually look at these slides, right, and try and identify these things. Oh, nothing left in there. And I want to stop my recording. How do I do that?